Hi everyone, my name is Lindsay Wilson, and today we're going to be talking about the modes of prokaryotic gene regulation, specifically negative repressible control. Let's first begin by explaining prokaryotic gene regulation. It's basically the way in which proteins, transcription factors, and other molecules control the expression of genes in a prokaryotic organism. These various molecules found within the cell can interact with each other and the DNA to either promote or inhibit mRNA transcription. There are two categories of regulatory mechanisms for transcription. The first is positive versus negative control. A positive control mechanism involves a protein called an activator that binds to DNA and increases the rate of transcription. Negative control mechanisms involve repressor proteins that actually prevent transcription from taking place when they bind to the DNA. The other category is inducible versus repressible control. During inducible control, transcription is usually off, but is then turned on when a small molecule or effector molecule binds to the regulatory protein. Repressible control is the opposite. Transcription is normally on, but the binding of a small molecule to the regulatory protein results in transcription turning off. It is important to remember that while positive and negative control deals with the proteins that bind to the DNA, inducible versus repressible controls deal with the small molecules that bind to the regulatory proteins themselves. When you combine both of these categories together, you end up with 4 different modes of gene regulation: negative inducible, positive inducible, negative repressible, and positive repressible. We're going to look specifically at the negative repressible mechanism. Let's break down the term negative repressible. The negative part means that there is a protein that is capable of inhibiting transcription, and the repressible part means that the presence of a small molecule will prompt the protein to turn transcription off. Here's a diagram of an operon to show how this process works. First, a regulatory gene or repressor gene upstream is transcribed and translated into a repressor protein. Downstream, there's a promoter region that indicates the site where RNA polymerase should start transcription. There's also an operator region where the repressor protein can attach. In the absence of a small molecule, however, the repressor is inactive. It is unable to bind to the operator because its shape does not fit onto the DNA. Since the repressor is not blocking the path of transcription, the RNA polymerase is able to bind to the promoter region and transcribe the structural gene sequences, which will then be translated into proteins. If the small molecule is present, though, they will bind to the repressor protein, causing a conformational change in its shape. Now, the repressor can bind to the operator, creating a blockage on the DNA. The RNA polymerase can no longer bind to the promoter, and transcription will not proceed. To help you understand the negative repressible mechanism, we'll use a SpongeBob analogy. Let's imagine SpongeBob is working hard on writing a paper for Mrs. Puff in boating school. The process of writing is analogous to transcription, and SpongeBob is like the RNA polymerase molecule, creating a wonderful paper rather than mRNA. As you might know, SpongeBob has a next-door neighbor named Squidward. He is normally at home alone where he is quiet and is like the inactive repressor protein. When Squidward has his clarinet, however, he likes to play very loudly with the windows open. This unpleasant noise is distracting to SpongeBob and prevents him from writing his boating paper. The clarinet acts as a small molecule that causes Squidward to inhibit SpongeBob's work. Thus, SpongeBob is only productive when Squidward does not have his clarinet. Here's a list of my sources, and that concludes this video on the negative repressor mechanism of prokaryotic gene regulation. Thanks for watching.